Hello guys, welcome back to A to Z Dentistry, a channel which features easy explanation of various medical and dentistry related topics. And our today's topic is endodontic biofilms. So without any further delay, let's get started. Starting with the definition. So, a biofilm is defined as a community of microcolonies of microorganisms in an aqueous solution that is surrounded by a matrix made up of glycocalyx, which also attaches the bacterial cells to a solid substratum. So, biofilm is basically a community of microcolonies of various microorganisms, right? They are suspended in an aqueous solution, and all these colonies are surrounded by a matrix which is made up of glycocalyx, right? And this biofilms serves a very important feature. So basically what it does is that it attaches the bacteria to a solid substratum, right? The tooth basically. So here in this picture we can see there are various bacteria, that there are microcolonies of bacteria. Some are spherical, some are rod shaped bacteria, and these are surrounded by a layer of glycocalyx, right? And all these are attached to the substratum, that is the tooth, via the glycocalyx layer. Moving on towards the composition of the biofilm. So, biofilm basically consists of 15% of the microorganisms and 85% of the extracellular polymeric substances, also known as EPS. Now, these EPS are basically produced by the microorganisms, right? And the majority content of this EPS is basically water, which accounts for approximately 97%. Then we have 1 to 2% of proteins and enzymes, right? And also polysaccharides constitute about 1 to 2%. Then less than 1% is DNA and RNA, right? So here it is clear that water makes the majority of the biofilm, and it is also responsible for the flow of nutrients inside the biofilm matrix. So these nutrients are basically derived from the saliva itself, and because of the large quantity of water which is present within the biofilm, there is easy flow of nutrients within the biofilm from the saliva. Moving on to stages in the development of the biofilm. So there are 4 stages, right, which contribute to the development of biofilm. The first one is formation of the conditioning layer, followed by bacterial cell attachment. Now these bacteria are planktonic bacteria specifically. So now what is a planktonic bacteria? Basically these are free floating microorganisms that live suspended in an aquatic environment, right, or in an aqueous solution. So as we have studied previously, this biofilm is basically an aqueous solution, right? So these bacteria are freely suspended within the aqueous solution of the biofilm. The third stage is the stage of detachment, also known as the seed dispersal stage or the seeding dispersal stage. And the last one is bacterial growth and expansion of the biofilm. So this is a schematic representation of the various stages in development of a biofilm, right? So in the first stage we can see there is attachment of the planktonic bacteria to the surface of the tooth. Now this is a reversible stage, right? The next stage is irreversible attachment and formation of the extracellular matrix. So this extracellular matrix basically protects the bacteria from the enzymatic action of saliva. The third stage is production of EPS, that is extracellular polymeric substances, by the bacteria, and formation of the microcolony. Now this EPS consists of 97% water, then 2% we have proteins and various enzymes, and less than 1% of DNA and RNA. And this EPS is secreted by the bacteria itself, which are present within the biofilm. And the fourth stage is the maturation of the biofilm, right? There is increased cell density and there is intercellular signaling which is happening at this stage, right? And the last stage is the dispersal stage or the seeding stage, right? Now here we can see there is detachment and dispersal of the planktonic cells or the free floating cells. Moving on to the most important part, that is the classification of biofilms, right? So it is classified into 4 major types. The first one is the intracanal biofilms, second is extraradicular, then we have periapical biofilms, and the fourth one is biomaterial centered infections. So now we will be studying each type of biofilm in a little bit of detail. So starting with intracanal microbial biofilms, right? So these are biofilms that are formed on the radicular dentine in an endodontically infected tooth, right? So when there is an endodontic infection going on, these biofilms are basically formed on the radicular part of dentine, right? So here in this picture we can see there is a formation of biofilm on the radicular dentine. So in this biofilm there are distinct types of bacterial colonies which are present, but E. faecalis is the major bacteria, and it is responsible for one of the most therapy resistant and prevalent endodontic biofilms, right? So this E. faecalis, as we know, is a very notorious organism, and it is responsible for majority of endodontic infections, right, which are very difficult to treat. So in this type of biofilms that are intracanal biofilms, majority of the time we can encounter this E. faecalis, and it is therapy resistant. So our next type of biofilm is extraradicular microbial biofilm, right? So as the name indicates, it will be formed on the extraradicular surface of the tooth, right? So these root surface biofilms are those that are formed on the cemental surface around the apex of the tooth, and this tooth is an endodontically infected tooth. So in this picture we can see there is a formation of biofilm on the cemental surface of the tooth, right? Moving on towards periapical microbial biofilms. So here, as the name indicates, it will form around the apex of the tooth, right? So these are isolated biofilms which are independent of the internal or the external surface of the root canal, right? These are found on the periapical region. So there are two major bacteria which are responsible and which mainly contribute towards development of these biofilms, that is actinomyces and P. propionicium, right? So now these lesions are usually very resistant to the endodontic therapy because of these two microbial organisms. Moving on towards our last type of biofilm, that is biomaterial centered infection. So these kind of biofilms occur when bacteria adhere to an artificial biomaterial surface and form the biofilm on these structures, right? A very good example would be an implant surface, right? So these biofilms are formed on the artificial biomaterial surface of the tooth. Now it is also known as the foreign body centered biofilm because it is not forming on the root, it is forming on an artificial surface. For example, in a post, right, in a post it can form, or in a screw, implant screw, it can form. So these type of biofilms are known as biomaterial centered biofilms or biomaterial centered infection or foreign body centered biofilm. Moving on towards the significance of endodontic biofilms. Now we need to know about these endodontic biofilms because there are certain reasons, right? It is very important that we understand a biofilm in order to successfully treat an tooth, right? So basically these are therapeutically significant as they are one of the basic survival methods which are employed by bacteria in terms of starvation. These are mainly responsible for endodontic failures. Now we have seen that there are certain organisms, right, like actinomyces, which are responsible for failure of endodontic treatments, and one of the major reasons why treatments fail is because of this formation of biofilms. Now these biofilms basically help the bacteria to survive because they have certain qualities, right? There are certain advantages which are provided by the biofilm to the bacteria. The first one is the ability to protect the bacteria from the environment. So as we know that saliva naturally contains various enzymes like lysozymes, it has antibacterial action, right? So what it will do, it will kill this bacteria, right, eventually. So but when there is a biofilm, it's like a protection around the bacterial surface. So its main function or the crucial function is it protects the bacteria from the environment, right? It has the ability to entrap the nutrients for the growth of microbial constituents. So we have discussed previously that because of the large amount of aqueous content of the biofilm, there is diffusion of nutrients, right, within the biofilm. So what happens is the biofilm basically takes up nutrients from the saliva, and in turn it feeds the bacteria which are present within them. So eventually there is growth of bacteria. Then they offer a safe environment for the exchange of genetic material among the constituent bacterial colonies. Now it's a very safe and protective environment for the bacteria, right? Because they are fully covered by this glycocalyx, right, of the biofilm itself. So it provides a very safe and secure environment, and it also promotes the genetic material exchange within the bacteria, which is responsible ultimately for the resistance, development of resistance within the bacteria itself. Then last one is the biofilm structure offers an inherent resistance to antimicrobial agents such as endodontic irrigants and intracanal medicaments. So basically it offers an resistance to the antimicrobial agents. Why? Because we have studied that there is an exchange of genetic material, right? So the resistant genes which are present in one bacteria are then given to another bacteria, right, through conjugation or through quorum sensing, right? So these are the two mechanisms. Now what happens is, because of these processes, various antimicrobial agents do not effectively work on this biofilms, right? So it offers a structure which is inherently resistant to a variety of drugs, endodontic irrigants and intracanal medicaments. Moving on towards antimicrobial resistance of the biofilm, right? So how this antimicrobial resistance is possible within a biofilm? So the first one is there is a slow penetration of antimicrobial agents, right? The EPS, extracellular polysaccharide structure in the biofilms, retards the diffusion of antimicrobial agent, right? So it's like a very protective covering around the bacterial colonies, and therefore the diffusion of antimicrobial agents is greatly reduced because of that. Then there is enhanced tolerance to antimicrobials, higher density of the cells, right? So because of this coral sensing and all, there is increased density of cells in a particular area, right? And because of that, the antimicrobials cannot work effectively. There are more number of bacteria as compared to the amount of drug or antimicrobial agent that we are using. Then arrangement of the cells. Microorganisms interiorly are protected from medicaments that act on microorganisms in the biofilm's periphery. Now most of these intracanal medicaments are very effective in killing the bacteria which are present at the periphery of the film, but the bacteria which are towards the core or the center are usually spared, right? Then growth rate of the cells in the biofilm. There is reduced growth rate and nutrition requirement of the resistant cells within the uh, uh, biofilm, right? Now this is because again there is transfer of genetic material happening, right? So because of that, the growth rate and nutrition requirement is greatly less for some of the bacteria, right, which are inherently resistant to the medicaments. So guys, that was all about endodontic biofilms, right? I hope you understood the topic very well. Thank you for listening to this video. Kindly consider subscribing to my channel for more such videos. Bye bye, take care.